In this lesson, we're going to learn more about enzymes and about their specific role, as well as the different regions on an enzyme, including active site, substrate, and allosteric site. Before we talk about the enzymes, I want to talk about why enzymes are so important. And one of those is a disease called PKU, or phenylketonuria. And this is a gene defect. And so there's basically a typo in the DNA. So that gene is never transcribed into the RNA, and so the appropriate protein is never translated from the RNA in the ribosome. The enzyme that's missing makes it impossible for that person to break down phenylalanine. And phenylalanine is found in so many different foods, and it's also released from aspartame or NutraSweet when it's digested. And so if a person cannot break down phenylalanine in the body, it can lead to some very serious consequences. So this is one of the several tests that newborns undergo to make sure they don't have PKU. And the reason for this is because if they don't test for it and the infant is missing that gene to break down phenylalanine, by the time they realized that there was a problem, the damage would have been done, because the inability to break this down can cause brain damage and developmental delays. And so they want to test every baby for it and make sure that if the baby is missing that gene, they can prevent that damage from occurring, because once it's done, it can't be undone. So why do we need enzymes in the first place? I mean, enzymes are just basically a protein with a special job. And the reason we need them is because, for example, the enzyme used in creating amino acids in our body, and without it, the reaction takes 78 million years. So if this is an amino acid that's essential to life, we've got a problem. And so we need enzymes to help speed up this reaction significantly so that we can get the things we need either formed or broken down in time so that we can survive. Another example is the biosynthesis of hemoglobin. Without the enzyme, the reaction takes 2.3 billion years. Given the role of hemoglobin in the transport of oxygen, this seems to be a little bit incompatible with life. And so those enzymes are absolutely essential. Now, missing some enzymes can have serious consequences. Missing others can be more annoying than deadly. For example, someone who's missing lactase or doesn't produce enough of the enzymes that break down lactose may need to watch their diet or deal with some uncomfortable side effects, but they're going to survive, whereas other enzymes are critical to life. So we've already talked about catalysts and their role in speeding up reactions, and enzymes are just biological catalysts. They lower the activation energy, just like the catalysts we saw in other reactions, and they're not consumed in the reaction. So we have an enzyme, we use it, the reactants come in, the reaction occurs, the products leave, and then the enzyme can be used over and over again. So it's there at the beginning of the reaction, and it's there at the end of the reaction. Let's look at an example to identify the catalyst. Now, this one is obviously not an actual enzyme, but it makes it a little bit simpler to look at the formulas than an actual enzyme reaction. Remember, what we're looking for is something that's present in the beginning and at the end, because that's the job of an enzyme. So when I look at my reaction, I see chlorine is present at the beginning. It's also present at the end. That's a good sign. I see O3 is present at the beginning, but it doesn't come back, so that can't be a catalyst. ClO is produced, but then it's consumed, so that's not a possibility. That would actually be something called an intermediate. O2 is produced, so it's not there in the beginning, so that's not a catalyst. And oxygen is just there in the middle. So Cl is the catalyst for this reaction. So as we talked about proteins having very specific needs for structure to maintain their function, enzymes are the same way. And so the proteins structure, primary, secondary, tertiary, and quaternary, all of that we talked about for proteins also applies to enzymes. Enzymes just have a specific job, and that's as biological catalyst. So we talked about lactase catalyzing the hydrolysis of lactose. Pepsin, which is in the stomach, catalyzes the decomposition of proteins into amino acids. And so we find pepsin in our stomach, and it works in a very acidic environment of our stomach, and it may not function well in a more neutral environment, such as in our saliva. So enzymes will function differently at different pHs and different temperature, depending on their preferred conditions. This reaction shows the first step of glycolysis. What is the enzyme for this reaction? So the correct answer is actually hexokinase, because we see ATP goes in, ADP comes out, and this is actually just present in the reaction. Another thing you can look for to identify them, if you haven't picked up on this already, is things like sucrase breaks down sucrose, lactase breaks down lactose. So that ASE ending is a good sign that you're dealing with an enzyme. Some enzymes, such as pepsin, use an older naming system, but many of them will use the newer ASE ending. Let's look at some of the basic places on an enzyme. So the first thing we have is our active site, and this is actually where the molecule comes in. So if we were dealing with this, let's say this is sucrase, okay? Then this would be sucrose coming in to interact with the sucrase, and so that the sucrose could break down. This whole thing is considered the enzyme, so the sucrose comes into the active site. There's also something called the allosteric site, and we'll talk about its role in just a minute. But notice that it is a site that can interact with other molecules, but it's not where our substrate or that main compound interacts. Let's look at the basic pathway for an substrate and its enzyme. So if we look at this, the substrate here is in green, so that's our molecule that needs to be broken apart. We have our active site of the enzyme where the substrate is going to interact with it. As the enzyme accepts that substrate, it changes slightly, kind of adjusts so that it can accept that substrate. And then we see the bond breaking between the two pieces of that particular substrate, and then the products leave separately from the enzyme. And now our enzyme is ready to go again and can repeat this cycle many, many times. And so what we see here is our substrate entering the active site of the enzyme substrate complex, the enzyme products complex, and the products leaving the active site of the enzyme. Based on the shape of the enzyme and the substrate, identify the allosteric site. So the allosteric site is B. Note that the shape of the substrate is very similar to the shape of A, and so this would be our active site where the substrate is going to interact, and this is the allosteric site.